Today we discuss very important topic, that is the nucleic acid. What is nucleic acid? You know that the nucleic acid are of two type. Nucleic acid are of two type. One is the DNA, another is the RNA. This is called deoxyribonucleic acid and this is called ribonucleic acid. The DNA is a large biomicromolecules and its formation or take place by the process of polymerization. So we can say that it is a polymer, and the monomeric unit of this polymer is the nucleotide. So today we discuss about the structure and component of DNA and RNA. So now first we discuss about the DNA. What is the DNA? DNA is a large micromolecules and it is made up of three basic components, or it is a large micromolecules, or it is a trimolecular structure, and its repeated unit that is called the nucleotide, and each nucleotide is made up of three different type of component. First is a pentose sugar, means 5 carbon molecules. This is the ring structure, and basically in the case of DNA, pentose sugar is a deoxyribose sugar. Its second component is a base, a nitrogenous bases, and these are also two type. One is the purine, another is the pyrimidine. Purine are double ring structure, whereas pyrimidine is the single ring structure. They are also two type. Purine are two type. One is adenosine, that can be represented by A, and next is the guanosine, that is represented by G. Same, pyrimidine is also two type. One is the cytosine, another is the thymine. The third basic component of nucleotides, that is a phosphoric acid. You know that the nucleotide is also a large macromolecules, is a large molecules. Its formation or take place in two steps. First, when a pentose sugar, this is the sugar, link with a nitrogenous base, either the A, either the G, either the C, either the T, then a dimolecular structure are formed. Here the bond is N-glycosidic bond, and finally a dimolecular structure is formed. This is called nucleoside. This is the first step of the formation of DNA. There are 4 steps. After that, a complete molecules of DNA are formed. In second, when a sugar and nitrogenous base, that is called the nucleoside, joint are bonded with the phosphate group by a specific bond, this is called the ester bond and form trimolecular structure is called nucleotide. You know that these nucleotides are the monomer of polynucleotide. Now polymerization are take place and many nucleotides are joined together by diester bond like this P. This is the phosphate group joint is with sugar and then nitrogen. Once again, this is the second nucleotide and both nucleotide are joined with a phosphate group and form diester bond, and this process is continue like this P. This is the sugar, this is the nitrogen, and this is called the polynucleotide chain. In a reference to DNA, it is not a single strand DNA, it is a double strand DNA. So we can say in DNA, two nucleotide chain join together in the anti-parallel motion. If a chain can be start from T5 to 3. This is also very important. What indicate 5 and what indicate 3? Actual में this show the moiety of the hydroxy group in a sugar, or in other words, when two nucleotide chain join together, then the phosphate group, this bonding are take place by 3 carbon, then this bonding are take place by the 5 carbon. So where the nucleotide chain is start, here a carbon is vacant. Here is no double bond, diester bond are take place. So this represented by 5 carbon. This is the moiety where the polynucleotide chain are formed. And in last, here also in sugar 3 carbon is vacant. So this is called, this can be represented by 5:3 moiety are the direction. And when two nucleotide chain are linked together by hydrogen bond between the nitrogenous base, like here this is N, this is N, this is N, and this is N. That is the sulfur of sugar. This is the sugar. This is the sugar that can be joined together by the phosphate group. So when two nucleotide chain are joined together, then a polynucleotide chain are formed. This structure is now called the DNA, deoxyribonucleic acid, because in DNA two like this polynucleotide chain can be arranged in a anti-polarity direction. We know that always the polymerization are take place in this manner, 5:3. So this is the 5 and this is the 3, and you know that this is the anti polynucleotide chain, so it is the 3 and that ends can be so the 5. And now this structure is called DNA. So this is the detailed structure of DNA or the nucleic acid. Now we discuss about the RNA. There is so much similarity between DNA and RNA. RNA is also made up of 3 basic components. Here first is the sugar. In RNA the sugar is ribose. Second is the nitrogenous base. In RNA also four different type of 2-purine and 2-pyrimidine nitrogenous base are found, but in reference to pyrimidine, the purine base is same as DNA, but pyrimidine base differ here. Instead of the T, here uracil are found. Instead of thymine, uracil are found, whereas its second component are the cytosine. And third component, that is the phosphate group, are same as DNA. And the structure of RNA is a single strand, so ultimately like this single polynucleotide chain are formed the molecules of ribonucleic acid or RNA. You know that in a reference to animal or in reference to living thing, the RNA are of three type. One is the mRNA, second is the tRNA, and last one is the rRNA. They can perform the different function. You can study in 12th standard, but in brief you know that this is the messenger RNA. The mRNA are formed when the transcription of DNA helix are take place, or in other words we can say the message of DNA are converted in the form of RNA, and this is called messenger RNA. Second is the tRNA. It is also called the adapter molecules because it have two different sides. One is the amino acid acceptor and another is the anticodon site. So it helpful for the process of translation or the process of protein synthesis. And the rRNA, or it is also called the ribosomal RNA, it is the main component that initiate or involve in the process of protein